I'd like to thank all the organizers for this wonderful opportunity to share some of my work. So I've just recently started a lab at the Simon Center in NCBS. So let me just make this. Okay, yeah. So I've just recently started my lab, just about 2 or 3 months at the Simon Center at NCBS. And what we do is a combination of experiments and theory. So my lab is doing both currently, and our sort of broad questions that we want to answer are fundamental principles of cellular proliferation and how it is controlled primarily in cancer cells. To give you a better sense of some of the questions that we are interested in, I want to immediately show you a very short movie that I generated towards the end of my postdoc in Boston. And even if it's a very short movie, there's some very interesting phenomena that I will point you to in this particular movie. So what you're seeing are the nuclei of two HEC293 cells. So these are some kind of human embryonic kidney cells. And you're seeing the nuclei because I've stained them for an H2B protein. So that's the color that you're seeing. And what I want to do is play this movie for you. Okay. So what you're going to see is that these two cells are now going to divide, right? So you've seen the first division. So you can call these two cells the sister cells or the daughter cells, right? Because they came from the same original mother, right? So the mother divided and gave you these two sister cells. Now, what I want you to do is observe when they next divide, okay? So they are separating, and so if you noticed in that movie, let me play that once again. So what you would have seen in this movie is that the cells, the sister cells, they divided almost at the same. I don't know why I'm not being able to play this movie once again, but what you would have seen is that those two sister cells, they divided essentially at the same time, even though they were separated by at least 3 or 4 cellular distances, right? So what this suggests to us, even in the short movie, is that there are these possible correlations that you might be able to observe when you see these single cells dividing over time. And that's in fact what we can do. So one of the interesting questions that we try to address is when you look at hundreds and hundreds, sometimes even thousands of these single cell division events in these time-lapse movies, you can start generating these lineages that look like this, right? So you saw the mother dividing into 2 daughters or sisters, and then they in turn divide, and you can start tracking the lineage relationship, right? So the cousins, first cousin, second cousin, third cousins, and so on. And if you define this time from when the cell was born to when the cell divides once again as the intermitotic time, or the IMT in short, you can start looking at correlations between various lineage groups, right? So between sisters, between mothers and daughters, and so on. And as I'm showing you here, this is one observation, which I alluded to in the previous movie. You can see that the sisters are very strongly correlated. And the questions we ask are, why do we see things like this? What can we infer about the way cells divide or cell cycle based upon these kinds of correlations? Okay? But these are not the only kinds of correlations that you can measure in these experiments. So what we also do are experiments where we add various kinds of drugs to these cancer cells. So they could be chemotherapy, there could be targeted agents. For example, here what I'm showing is a cisplatin treatment. And you can add this treatment and look at the data at various different points. So you can look at sisters that were born before drug treatment, sisters that were born after drug treatment. And instead of this intermitotic time correlations, you can ask a slightly different question after you've added drugs, and you can ask, what are the correlations in end fate of these cells? So the moment you add drugs, cells will either die, as I've shown you in this figure, or they will survive this cisplatin treatment, right? So they will develop some kind of resistance to the treatment. And you can ask questions like, what kind of lineage correlations do you see in the development of resistance or in the survival of these cells? Okay, and what do we learn about the nature of cancer cell drug resistance evolution? Is it a Lamarckian process? Is it a Darwinian process? These are the kinds of questions we try to understand by doing these experiments and building theoretical frameworks. And finally, of course, what we also do is we add reporters of various proteins to these single cells. So in this particular example, you're seeing a reporter that it's a fluorescence reporter that reports on the p53 protein dynamics, right? And you can see that we've built these lineages here. So a cell divides, and then divides once again here, divides here. You see those lineage correlation relationships, and you can also track the protein dynamics as a function of time. So that's the curves that you see over here, right? And you can again look at cells that have different end fates. So the red curves are cells that died, the blue curves are cells that survived. And you can start asking what kind of correlations are there in the dynamics. Can you build sort of inference frameworks of using stochastic models to understand the underlying principles again of drug resistance? How do you control the proliferation of these cells using mathematical models and so on? Okay, so that's sort of broadly what we are interested in as a group. So today what I'm going to do is give you one example of this kind of work. So an example drawn from this top level kind of picture, where we are looking at correlations in the intermitotic times, okay? And the rest, hopefully someday in the future, I'll talk more about the rest of the work, okay? So it turns out that if I start looking at these kinds of intermitotic time correlations, there's a very interesting phenomenon that we start observing. And this is, we did this in a particular cell line called HCT116, which is a cancer cell line. It's a colon cancer cell line. But this is, in fact, it has been seen in many other cells as well. So the observation is the following. It's that if you look at the mother-daughter correlations in intermitotic times, you see that basically there is no correlation. So rho here is the Pearson correlation, okay? So there's no significant correlation over here. If you look at the sisters, and again, this is the movie I was trying to allude to, you see a very strong correlation, so 0.73. Interestingly, as you can see here, the cousin correlations are quite large, and in fact are larger than the mother-daughter correlations. So if you pause for a moment, this may appear kind of surprising to you, right? Why is that? So if I go back once quickly, what it's saying is that mother and daughters, so mother would be this cell, the daughter would be this cell, right? So you're losing information on division times between mother and daughters, but somehow in cousins, that information is coming up again, right? So your cousins are more strongly correlated than mother-daughters. So this phenomenon was named the cousin-mother inequality in this beautiful paper in Nature from Natalie Balaban's group, right? And the question is, why does that happen? So the first question that we had asked in this paper, which is now published, you can go read it, is this phenomenon surprising, right? I mean, intuitively, it seems surprising to us, but is it really surprising? And what are the underlying assumptions going into a model that would tell us whether this phenomenon is even surprising or not, right? So I'm not going to go into too much detail of this aspect, but what we did is we built a simple stochastic model based on the kind of ideas people have currently in the field about how cell cycle is regulated. And this is represented here in the simple cartoon. So let's say a cell was born with very low amounts of a certain protein, right? I'm calling it this protein X, but this is usually thought to be some kind of cyclin-dependent kinase, or some people have even suggested that this is the ratio between cyclin-dependent kinases and P21, for instance, right? So these things that control how quickly the cell is going to divide. So let's say the cell is born with low amounts of this protein, which is denoted by this black line here, and that results in a longer lifetime for this particular cell, okay? But over the course of this lifetime, just because of random fluctuations in the protein levels due to stochastic production and degradation, let's say the level of that protein rises to some higher value. That's shown over here. So when this cell now divides, the daughter is given, so basically these high levels of that protein or the ratios of different proteins are transmitted to the daughter cell. And because this daughter cell is born with high amounts of this protein, it survives for a shorter time because it has higher cyclin-dependent kinases and so on. So it cycles faster through the cell cycle. So that's roughly the current understanding of how cell cycle is controlled. So interestingly, if you develop a stochastic model with these principles, what we showed is that you can't recapitulate this cousin-mother inequality in the kind of data that we actually observe. So that's shown over here. These are simulation results from our model, these box plots, and the dashed lines are 95% confidence intervals of the data. So what you see here mainly is that the cousin correlations, that's these blue box plots, these are always going to be smaller than the mother-daughter correlations, okay? So this kind of model doesn't explain the data, the cousin-mother inequality. So of course, the natural question is then what extra thing do we need to put into our models to be able to understand this kind of a phenomenon? So it turns out that again, in this paper from the Balaban group, they had suggested a toy model where the idea is that there is some kind of external deterministic control over the cell cycle, and they posited a circadian rhythm mediated control of the cell cycle that may give rise to these kinds of correlations. So what we decided to do is build a data-driven model to see whether all aspects of our data in this cancer cell line can be recapitulated with a circadian-driven cell cycle model. So to do that, the essential idea that I developed in this paper was the following, right? So imagine that you have some circadian proteins, right? So the circadian clock is essentially an autonomous clock which oscillates in every single cell of our body, even if you take it outside. And these circadian proteins oscillate with roughly a 24-hour time period, okay? So, and of course, that sets the phase of the clock, so that's what I'm showing you over here, okay? So this phase goes between 0 to 2 pi. So imagine a situation where the phase of the clock at the time a cell is born, imagine that that phase sets how quickly that cell is going to divide, okay? Now, if you've built a model like that, let me explain to you intuitively why you might expect cousin correlations that are stronger than mother-daughter correlations, right? So what's going to happen is the following. Each individual cell in your population is going to divide at slightly different times, okay? So there's an intrinsic heterogeneity in cell division times in any given cell line or any given population of cells. So that's represented here in this cartoon. So these cells divide with different times. Now, if you have that situation, what's going to happen is sister cells are going to be born at exactly the same time or exactly the same phase of the circadian clock, okay? Therefore, their rate of cell division in the model is going to be the same, okay? Whereas if you have cousins, so this cell and this cell, these are cousins, they are going to be born at roughly similar phases of this of the circadian clock, but not exactly the same phase as I'm showing you here, okay? What that will mean is that the rate of division of the cousin cells will be similar, but not the same. Okay. And as a result, you'll get some correlations among cousin cells. But the mother-daughter correlations will be set completely by the average cell division time, right? So it'll depend on how long these cell division times are. So depending on that time, the cells, the mothers and the daughters, could be born at very different phases of the circadian clock. And hence, their correlations could essentially be zero. Okay, so this is the model in a little bit more quantitative fashion. The way we introduce this model in the field is basically we are thinking of rates of division. So in the survival analysis literature, these are called hazard functions. Okay, so think of these as essentially time dependent extensions of your simple rate in a Poisson process, right? In a Poisson process, your rate is a constant as a function of time. Here, your rates are time dependent, as you can see over here. Okay, and the basic idea again is that these hazard functions for division depend on the phase of the circadian clock. Okay, so let's see if we put this model into sort of these into a simulation, into a Gillespie type simulation framework, where we are simulating the growth of these single cell populations using hazard functions that depend on the circadian phase. Can we recapitulate the data that we see? And that's what I'm showing you over here. Indeed, we can recapitulate this kind of cousin mother inequality. So again, what I'm showing you here are the mother-daughter correlations in this sort of pink box plot. These are the sister correlations, and these are the cousin correlations. And the dashed lines are the 95% confidence intervals directly from the data. And what you can see now is that almost quantitatively, we can recapitulate the fact that these cousin correlations are larger than the mother-daughter correlations. And in fact, we're getting the quantitative values also within the 95% confidence intervals.
Shayan, would you like to take two questions in between?
Yeah, absolutely. Sure. Go ahead.
Okay, so there is one question from Anne George. She's asking that, is there any significance to the position of cluster of points in the graph shown earlier? She's referring to 2 slides back.
Yeah, which one?
Yeah, so the question is there any significance to the position of cluster of points in the graphs shown here? I think she's referring to the upper graphs.
Yeah.
So
I'm not exactly sure what the question is. I mean, what you can do is you can generate confidence intervals and also look at the significance levels of these correlations. So here I've just given you these point estimates, but of course you can generate confidence intervals and significance values, so p values. So we did all of that, and all that data is there in the paper. And you can see those 95% confidence intervals over here as well. They are kind of large, but they track the trend pretty well. The fact that cousin correlations are larger than mother-daughter correlations.
Yeah.
So I hope that answers the question. Yeah.
So second question is that the intermitotic time need not be, I guess this is a comment. The intermitotic time need not be same for the 2 daughter cells?
Oh, absolutely. Yeah. So that's exactly what I'm showing you over here. So remember that this is a probabilistic framework, right? So these hazard functions are essentially rates of cell division. So in this model, what I'm saying is, depending on the phase of the clock at which these 2 cells are born, okay? So this is going to be identical in this model, right? Because the 2 sisters are born exactly at the same time. Okay, and that I'm saying is going to have the same phase of the clock. Now that will set the hazard function for division. So what it will do is it will make the rate of cell division faster depending on the phase of the clock. But that does not necessarily mean that these 2 cells will divide exactly at the same time, right? These are probabilities, right? These hazard functions, these are not deterministic quantities. So yes, and these are going to be very important in the rest of my talk, the fact that there is some kind of heterogeneity. And to follow up on that point, this is precisely what that heterogeneity is, right? So if you plot the division time distribution, you see that there is a kind of wide distribution over here. And what I'm showing you here is that the model can in fact recapitulate the entire distribution as well. So the histograms is directly from the experimental data. And this dashed line here with the confidence intervals, that is the results of this model, okay?
Shaon,
Yeah.
one question. So
Sure.
this PDF is actually the first passage time, right? Of the, I mean, this IMT is the first
Absolutely.
time. So you have first passage time distribution, right?
Absolutely, absolutely. You can definitely think of it as a first passage time distribution. Yes, absolutely. Yeah, absolutely.
Okay, thank you.
Yeah. And the other thing that I want to point out here is the fact that our model has only 4 free parameters, okay? What is the reason? The reason is because these distributions tend to be right skewed, okay? So these are not Gaussian distributions, these are skewed. So the moment you have a skew in your distribution, you require at least 3 parameters to describe them, right? You're the mean, variance, and your skew. So that's 3 parameters already for your distribution. And then with only 1 extra free parameter, we can recapitulate all these correlations as well, okay? So that was something that made us really happy, that we are sure that we are definitely not over parameterizing our model, okay? So I can't hear you. I could only hear it very vaguely.
Can we defer that to the end of the talk, since it's a short talk? Yeah.
Yeah. Okay. So the next question that we asked is, okay, so we've in this slide, we've shown that a model with a 24-hour oscillator can explain all of this data coming from our microscopy experiments. But we also know that a lot of the so-called ultradian rhythms, so for instance, NF-kappa B oscillations, Hess1, Hess7 oscillations, these almost always exist in mammalian cells, and they tend to have a time period of oscillation of the order of 3.5, 4, 5 hours, okay? And we know that these oscillations also can often influence how quickly cells divide. Okay? So the next question we asked is, can ultradian rhythms explain the data that we are observing? And in short, the answer is no. And the intuitive result, the reason behind that is shown in this cartoon over here. The idea is that if your oscillator is oscillating really rapidly, then what's going to happen is that cousin cells are going to be essentially born at random phases of the clock, right? If your oscillator was slow, right, it was oscillating with a 24-hour time period, the envelope of that circadian oscillation would have been changing in a much more gradual fashion. Hence, the cousins would have been born at roughly similar phases. But when the oscillations are this rapid, the cousins are essentially born at completely randomized phases of the circadian clock, right? And when you put that into your model, into our model, and simulate the results, what you see is exactly what we had expected from this intuitive picture. The cousin correlations are essentially 0, okay? So what I've shown you here is that the interplay between the heterogeneity over here, right? The fact that these division times are slightly different, which make the cousins, so the cousins are born at slightly different phases of the clock, right? So that interplay between the heterogeneity of cell cycle times, the circadian clock oscillation time period, and also the mean length of the cell cycle time, all of these together couple to generate these kinds of correlations in the data. So the next question we asked was, can we actually mess around with the heterogeneity in these in these times, right? And see whether we can still recapitulate all these correlations from the data. So to do that, what we did is we added cisplatin. So cisplatin is a chemotherapy which is known to generate more heterogeneous cell cycle times. So the experiment looks exactly like this. So we have these cells dividing, and then we add the cisplatin after about 2 days, and then we let the cells we track the cells under the microscope for another 3 days. Okay. So now the interesting thing, which is slightly different from our scenario before, is that we have multiple competing fates. Okay. What that means is your cells can now die because you've started adding cisplatin, or they can survive the treatment, or they can go ahead and divide. Okay. So you have these multiple fates that you can observe. So now, instead of just the division time distribution, we are also thinking of the times to apoptosis or times to death. Okay, so that's what I'm showing you over here. Oh, sorry. This what I'm showing you here is actually both the division times. And the reason I'm showing you this figure is to show you that as we had expected, after drug, the distribution is much wider, right? So remember, I had told you that we are introducing this drug to try and increase the heterogeneity in cell cycle times. And that's exactly what you see over here. That's distribution has become much wider as compared to the before drug scenario. And the question is, can we still recapitulate the correlations? But there's one problem, which is an interesting theoretical problem over here in trying to understand what exactly the cell cycle times are when you have these competing risks. And let me try and explain what that problem is, okay? Here's the problem. So imagine we're doing a thought experiment, okay? Where the division time distributions are generated from this red distribution over here, okay? The apoptosis time distributions are generated from this green distribution over here, okay? So those are the underlying distributions that are inherently part of the cell cycle and cell death times, okay? But what is going to happen because of the fact that you are not going to be able to observe both these events for any given cell, right? So these two events are mutually exclusive. So if a cell goes and divides, you will not observe it dying, and the other way around. So what that leads to is this phenomenon called competing risks, okay? What will happen is that the observed distributions that you see in the actual experiment will be highly biased towards the left. And the reason is very simple to understand. If the cell was going to divide with, let's say, a time drawn from the right tail of the distribution, so say somewhere here, and you've added drugs, it's very likely that the cell is actually going to die before it has the chance to divide. So what in reality, what you will see is you'll never be able to observe these right tails of the distribution, right? So what you're going to in fact see is this highly skewed distribution in reality. So we have this interesting challenge that given the experimental data that looks like this, we want to solve an inverse problem. So starting from here, we want to be able to figure out what the actual underlying distributions were so that we can start thinking about whether these real distributions can give rise to the correlations we observe. So to solve this problem, I developed this Bayesian strategy, and I'll just very quickly walk you through what the strategy is. So remember that we have these very strong correlations in sister times, okay? So that is something that is also needed to be included in the inference framework. So the way I went about doing this is T1 is the time for division of, let's say, sister 1, and T2 is the time for division or death, right, for sister 2. And what I did is I wrote down a joint density for these two sisters, okay? So this joint density of the two sisters depends on a set of parameters, theta, which describe the distribution of the hazard functions. This joint density can be written down using a very interesting theorem called class theorem as a product of a particular function called copula. So this accounts for sister correlations and the marginal densities of these two sisters. So in fact, it turns out that this is a completely general result. You can write down any arbitrary joint density function as the product of copulas and the marginal densities. So this allows you to separate the correlation structure from the marginal densities. So after writing down this joint density of just two sisters, I describe the entire data set as a product of sisters, right? So that gives me a likelihood function, and then you can do some kind of Markov chain Monte Carlo to infer what these theta values are, okay? And that is, the results are shown over here. As I had told you, the inferred distribution, again, remember this green dashed curves, this distribution of cell cycle times is not observed, but is inferred from this Bayesian procedure, right? And what you're seeing here is that this distribution is in fact very, very different from the observed distribution of cell cycle times. So the red histogram is the observed distribution, and the green distribution is what we've inferred from the data. And what we are suggesting, and this is why we call the paper hidden heterogeneity, when you're adding drugs and when you have multiple competing fates, it's not enough to say that this distribution is the correct distribution. And unfortunately, this is really what is done always in the field, okay? So really, what you need is a distribution that looks like this, okay? So what we can now do, given that we know what the underlying real distribution of cell cycle time and apoptosis times are, we can put this into, again, a similar sort of Gillespie type simulation framework and generate lineages of cells that are dividing. And we can start looking at the correlations that we generate in that simulation framework, right? And what I'm showing you here again is that even after adding the drugs, when you've generated all that heterogeneity, we can explain these correlations in sister correlations and cousin correlations in both division times and times to death using this kind of a model of the circadian clock controlling the cell cycle lengths, okay, or the cell cycle speed. So with that, I'm going to summarize because I've run over time. So maybe the final thing I'll say is that, of course, all of this suggests that the circadian clock is somehow controlling the speed of the cell cycle. Of course, now there are one or two other models in the literature that can also explain these correlations without invoking a circadian clock. So that's the very interesting situation we are in right now. There are about 3 models that explain these correlations without, 2 models that explain it without the circadian clock. So what we are doing in the lab currently is we are building experiments where we can look at mammalian cells. So we are in fact working with a stem cell system where, depending on how differentiated it is, we can actually induce the circadian clock oscillations versus get rid of the circadian clock oscillations. So with these 2 different systems, we are now looking at the correlations in the cell cycle times that are observed in the real data and trying to understand whether actually the circadian clock does indeed affect the cell cycle speed. Okay, so in summary, what I've tried to argue is that, in the least, the current understanding of cell fate control, based on these simple ideas of protein production and degradation and how they get split between daughter cells, passed on from mother to daughters, this cannot explain the correlations that we see in cell cycle times. Okay, how do we explain those correlations then? So we developed a model based on this terminology of hazard functions, which gives us a general framework of thinking of circadian gating of cell division and death. And we've shown that circadian clock, but not the ultradian clocks, might be driving the cell cycle speed, at least in terms of these correlations that we observe. And finally, I've tried to argue that when you add drugs to these cells such that you start inducing multiple different fates in these cells, you cannot simply go to the experimental data, look at it, and say that that is the distribution of cell cycle times. Okay, you really need to decouple this sort of competing risks effect that underlies these processes. So finally, my acknowledgements. All of this The work that I've shown you here was done while I was still a postdoc. Most of the experiments that I showed were done by Andrew, who was at that time a postdoc in Galit Lahav's lab. So we worked very closely together, and we had a great time with this paper. So Andrew is now a faculty at the University of Arizona. And then, of course, Franziska and Sahand, who were my advisors. The experiments that I did and the movie that I generated were all done in Sahan's lab. Thank you, everyone. And I would be happy to take any questions if there are questions. Thank you.
Thank you, Shayan, for the nice introductory talk on the topic. Yeah, so we'll start with the questions in the chat box. I guess you can also see them. Okay, there is a question from Kavya Shri. She's saying, let's say that they divided at the same time. I guess this is a follow-up from the last question, an earlier question. Yeah, so this is about the daughter cells. Let's say they did divide at the same time, then won't the mother-cousin correlations become more important than less?
The mother-cousin correlations, right? Yeah. So okay. So at least in this data set, we didn't see very strong mother cousin. So maybe what you're saying is mother and these things, right? So perhaps you could call this mother and granddaughter, right?
She's asking if the daughters divided at the same time, then how things will change.
If the daughters divide at exactly the same time,
yeah,
that's not going to change anything. What that would mean, so first of all, in the experiments, that is very rarely seen. We see a strong correlation, right? So the daughters are very strongly correlated, about 0.8 correlations. But that means that the two daughters are dividing at slightly different times, right? So this kind of heterogeneity almost always exists. And if there are rare cases where the two daughters divide exactly at the same time, that's still fine. So in that particular instance, the cousins in the model will be very strongly correlated. That's fine. But in the entire data set, that is rarely going to happen where the two sisters divide exactly at the same time, because these are inherently probabilistic descriptions of cell division. Yeah.
Okay. And the next question is from Aritri. Are there any factors that influence the ultradian rhythm periods? Can they be manipulated to increase or decrease correlations?
So again, I am not very familiar with how you could manipulate the ultradian clock. But as I sort of argued in this work, the ultradian clock doesn't seem to be controlling the cell cycle speed that much in terms of at least generating these correlations. But the question is very good. And this is in fact something that we are doing in the lab right now, which is we are trying to play around with the circadian clock. So not the ultradian clock, but the circadian clock. So there are small molecule inhibitors. So for instance, there's one particular small molecule inhibitor called KL001, which is very nice because at different concentrations of that small molecule inhibitor, you can actually continuously change the time period and amplitude of oscillations of the circadian clock. So we are adding that and trying to figure out, by changing the circadian clock amplitude and time period, can we actually control the cell cycle speed in cancer cells after we add particular chemotherapies, right? So the idea there becomes, can we actually use the circadian clock as a tool to control how fast cancer cells grow and die? So that's something we are doing there, not in ultradian clocks.
Okay, the next question is from Bhavakshi. How are the correlation coefficients evaluated from the theoretical models? Can you tell us about details of the approach or the mathematical steps to some extent?
Sure, absolutely. So that's a good question. And sorry, I didn't have time to go through that in much detail. So the basic idea in the mathematical model is we learn the basic underlying distribution. So this distribution, the histogram, is directly from the data. So let's say we are looking at only the cell divisions, right? So we can learn what that distribution is directly from the data. Now, once you know what that distribution is, it turns out that you can actually compute what the hazard function is. Okay, so there is a direct one-to-one relationship between a density function and a hazard function. Okay, so the hazard is given only by the density divided by the survival function. So once you know what the hazard function is, so let's say this is the hazard function that we learn from the data. In the model, what we do is we simulate single cells dividing over time. Okay, so let's say in the model we start with a single cell. We give the cell the hazard function that we've learned from the data. Okay, and that tells us that what is the chance that in the next certain amount of time the cell is going to divide. Okay, so essentially it's a Gillespie algorithm where we simulate single cells dividing as a function of time. Once we have these kind of single cell divisions in the simulation, we use a graph theoretical sort of framework to keep track of every single cell, and the nodes are basically every single cell's division times. So then we generate this entire lineage in a simulation, and from that lineage then you can sort of backtrack and say how much time it took for this cell to divide, for this cell to divide, and so on. And then we can just calculate the correlations in division times of sisters just from that simulation. Yeah, so that's essentially how it goes.
Yeah.
I have a question directly. Can you go to this formula that you wrote down, the copula and all that?
Oh, yeah, sure.
So there, this marginal densities of sister 1 and 2, are these your priors?
No, so basically this, all of this, right, goes into the likelihood. So in this particular treatment, I just set the priors to 1. So the likelihood function is just going to be a product over all these joint densities of all the sisters in this data set. So the data set is going to be comprised of, let's say, 1,000 cells, right? So what I do is those 1,000 cells, I divide into pairs of sisters because I know that the sister correlations are the strongest. So I'm basically ignoring all the higher order correlations among cousins and so on. So then I write down this joint density for these sisters, and then the likelihood function is just going to be a product of all these joint densities. So basically products of this right-hand side. Right, for all the cells. And then once I have that, essentially I could have maximized it and found out what these parameters theta are. But it turns out that when you have such a complicated system, I just could not maximize it using any maximizer or even find out the error bars on all these thetas, right? Because these gradient descent algorithms basically fail when your function is so complicated. So therefore, I basically did a Markov chain Monte Carlo by setting the prior to 1. So essentially my posterior and likelihood are essentially the same.
Okay.
There is
it is Bayesian, but actually it is a maximum likelihood than Bayesian, right? I mean, because the prior
it's yeah. So in the sense that the prior is set to 1, so it's basically a non-informative prior, but the likelihood I'm not maximizing, I'm still finding the entire distributions of these theta values. So the thetas are, let's say, the mean, median, and the mean variance and the skewness of those distributions. So those parameters I still can get entire distributions instead of point estimates. So in that sense, it is still Bayesian.
Okay.
So I'm not getting point estimates for these thetas.
Okay. Thank you.
There is a question from Shakuntala. She raised hands. Shakuntala, can you unmute yourself and go ahead?
Yes. Can you hear me now?
Yeah, I can hear you. Yeah.
Okay. Shaon, it was a very clear talk.
Thank you.
I was just thinking that how far down the generations the correlations are preserved.
Yeah, so that's an excellent question. The thing is, we are trying to look into this using a particular machine called the mother machine, which are microfluidic devices where you can track many generations. The problem with the sort of doing what you asked in the setup that we had here is the following. So you can see this field of view, right? What happens is once these things start dividing, the field of view gets cluttered with many cells, and it becomes very hard to track these lineages once there are too many cells in the field of view. So in these kinds of experiments, it's very hard to go many generations in. So we didn't have, at least in this data set, we didn't have enough generations in time to be able to properly answer the question that you are asking. So for that, we are using this mother machine, where this problem of cells crowding the environment is got rid of. So hopefully we'll be able to answer those kinds of questions sometime in the future. But that's a very good question, yeah.
Okay, Shaon, there are two more questions.
Sure.
Basil is asking, if I understand it right, you have considered the cell doubling time of cells lesser than the period of circadian rhythm. What happens
Yes.
if it is the opposite?
That is a good question. The experiments, the question is absolutely correct. The HCT116 cell line has an average division time of about 16 hours, which you can see over here. If it is more than the circadian clock, I'd have to think a little bit of what we would expect. We could definitely put that into the model and check. I've not done that. The reason we didn't do that is it's hard to get cells that have such long division times. But one thing we did do, which is when we added the drug, right? What you notice is that the average division time here, so this distribution is our underlying distribution of cell cycle times. So if you take the average of this does become greater than 24 hours, right? So this is kind of a weird scenario where you've added the drugs, which has resulted in very long cell cycle times. But there also, remember, we saw these kinds of correlations. So you can see the sister correlations, they have gone down. So that's the interesting bit. The correlations clearly have gone down. It's about 0.4 now, the sister correlations, but they still exist. But that's actually a very good question. So if you keep increasing the width of that distribution, you will expect the correlations to go down. And that's sort of obvious from this kind of a picture, right? If you make these division times very different, the correlations will start going down. And that's exactly what we see over here. Yeah.
Okay, the next question is so this is a follow-up question regarding the correlation coefficients. The simulations were carried out, which indicates that the time for each division was randomized. Does that mean that this randomization doesn't affect the final value of the coefficient as compared to the definite division times of the experiment? It's a long question. Can you read it in the chat? Maybe you'll understand it better.
Sorry. So let me see.
Yeah,
if I stop sharing, I might be able to.
Yeah, so just go to chat.
So now in the chat, okay.
Second last question is a little long.
So it's this one, right? Follow-up question regarding the correlation coefficient.
Yeah, it is. No. It is the second last. Bhashakshi Punya. Second last.
Yeah. Okay. The final value of the coefficient. So I'm not sure I understand the question completely, but the time for each division was not randomized in the sense that these are uniform random variables, right? The sisters are still going to be correlated because they have similar hazard functions. So the idea, remember, is, in the model at least, is that depending on the phase of the clock oscillation, the cells are going to have hazard functions. So if let's say the cells, two sisters were born at the peak of the circadian clock, they have a larger hazard function, versus if the cells were born at the trough of that circadian oscillation, they have smaller hazard functions. What that means is, even though the division of these two cells are probabilistic in nature, it's more likely that they will divide faster than the two cells that were born at a different phase of the clock. Okay. So that will set the correlations, and they're not going to destroy the correlations. So I hope I explained that.
So Shaon, I think, if I understand correctly, not all memories are lost in this randomization. The memory is being carried forward by the hazard function.
Exactly. The memory is carried by the hazard function in a circadian phase dependent manner. Exactly. Yeah.
Okay, thank you.
Right. Absolutely. That's a very nice way to put it. Yeah.
And the last question is that, I think this is again a follow-up question. In the real life usage, there are multiple cells undergoing the division. So how can you differentiate when there are more than two cousins from two parents in a particular time frame?
Yeah. So in the real life scenario, when you do these experiments, right, you can very easily track these things.
So Shaon, you have to share your screen.
Yeah. I forgot that it's not shared.
I think that's this.
Yeah. So here, let me share the screen. Let me see. So can you see the presentation now? Yeah. So if I play this movie, notice that we can actually track every single cell. So let this movie play out fully. You'll see some more division. So what we do in the lab is we generate these kinds of movies, and we can basically track every single cell. So you see these two are sisters and these two are sisters. So the identity of these guys are not going to be lost till your thing becomes really packed. So notice these two are sisters, these two are sisters. Now you know that this one and this one is a cousin, right? And now you see that this one has now divided. So what that means is that this guy and this guy are second cousins, right? So this cell and this cell is a first cousin. This cell and this cell are second cousins. So you can maintain the identity of every single cell in these kinds of experiments. So I hope that answers the question.
Okay. Shaon, I had one small question.
Yeah,
so,
sure.
Are there any independent signature of this circadian clocks so far as cell division goes?
Independent.
Like, for example, in these cases, you had sort of assumed that there is a circadian clock which is controlling the gaps of cell division.
Absolutely.
So is that an assumption, or there is some
so that's a very good question. In this particular paper, it was an assumption, but this was based on prior information from other groups and other labs, where they had suggested that the circadian clock controls the cell cycle. So this is, for instance, there are famous papers where people have shown that in these sort of network pictures, the circadian proteins actually go and bind to cell cycle proteins, right? So there is a lot of information, at least at the molecular level, that the circadian clock proteins directly interact with the cell cycle proteins. Now, what is the result of that is not known, right? And so in that sense, we made an assumption based on the prior experimental literature. But we can directly observe that, and this is in fact something that my lab is doing right now. We can put reporters of the circadian clock, so fluorescence oscillations that you can see under the microscope of the circadian clock. And then you can develop frameworks again based on the hazard function. So this is in fact why I introduced the hazard function in this work, is because hazard functions are very good for doing inference. So what we are trying to do now is directly by observing the circadian clock oscillations in real time, we are trying to understand whether the cell divisions are actually being affected by the circadian clock oscillations. So if we can do that, that will no longer be an assumption, because there we directly report on the circadian clock proteins. So does that make sense, Anirban?
Yeah, sure.
So I have a related question.
Sure.
Related to anyone's question. Shaon, the circadian clock is responsible for this. Now that has to somehow coordinate with those clocks which decide the sequence in the cell cycle, like how it goes from interphase to mitotic to cytokinesis and so on. So,
Yeah,
so that must be coupling with them because it cannot divide before those stages are complete, right?
absolutely, absolutely correct. Yeah. So that is an excellent question. And again, what we are doing right now, so we have these, we are getting some cells from our collaborator. So these cells have reporters for the cell cycle. So there are particular proteins that can be treated as reporters of the cell cycle. So whether the cell is in the G1 phase or S or G2M and so on. And also these cells have reporters for the circadian clock. So we can in real time see how exactly the circadian clock is oscillating, how the cell cycle phases are progressing, and based on those time-lapse microscopy movies, we can then start generating these inference techniques of how precisely the circadian clock is coupled to the cell cycle clock. So that's in fact something that some of my students are currently doing as we speak.
Oh,
Yeah,
okay. Okay.
yeah. Sorry, Anirban, I think we can't hear you. You're muted.
Yeah, sorry. Yeah, so this is a question from Deepjyoti Das. Depending on the clock frequency, can there be any optima in the sister correlation coefficients?
Yeah, that's a very good question. That's a good question. We are trying to look at that. So I think what will happen is it'll depend on the clock frequency, the heterogeneity in the cell cycle times, and also the average cell cycle time. So all of these three things together, I think will set the correlation coefficients. But definitely for some values of the circadian clock, you should be able to see an optimum, definitely. Yeah.
Okay, looks like there are no more questions. I mean, this has generated so many questions. Great. Thank you, Shayan. Yeah. I don't see any more questions in the chat or nobody has raised hands. I think questions are done. Yeah. Okay, it doesn't look like. Yeah. So thank you, Shayan.